see that. And can you hear me, everyone?
Bring the mic closer.
Mic up.
Mic up.
How's that? All right, thanks everyone for coming, or I guess there's no other session, so thanks for listening to me talk about the problems that I associate with my dissertation on this plant, Claytonia lanceolata. And for those of you that don't study Claytonia or work in the Caryophyllales, Claytonia is a very diverse genus, although it's not very well understood. And it's in the family Montiaceae, which is the bulk of the diversity in what used to be treated as Portulacaceae, now Portulacini, outside of the massive cacti clade. About 27 species in the monograph. I would argue there's at least twice as many of that in Western North America. And many species occur in sympatry, so you'll find multiple species at any given location, and therefore they do hybridize, and they don't take their chromosomes seriously at all. Pretty much every species has some polyploid antiploid series. And just to give you a sense of the diversity of parenation structures, we have these palatable tuberous species that I study, and also these more like carrots, coticose perennials, and various rhizomatous perennials. This one has a thin, woody rhizome, and this one with thick, fleshy rhizomes. And also to give you a sense of the magnitude of the problem I'm studying, this is the distribution of the Claytonia lanceolata species complex, and an illustration from the original description. And outside of this illustration, there's really not a lot of information in the protologue, so this is effectively what I'm looking for. Fortunately for me, there is a ton of diversity in California that's been unaccounted for, so I narrowed the scope of my dissertation to focus really on California. And fortunate for you, there's a ton of diversity down here that I don't have to talk about, so I'll just be talking about a simple story with 3 species in Northern California. So this is Claytonia lanceolata, and this is the closest thing that we have in California to what you can call, in the strict sense, lanceolata. I call it in the broad sense because I don't really have a good understanding of the distribution of that species. It really does extend all the way up to Canada and gets pretty close to Mexico. And I'm in Southern California, and they're all flowering at the same time for 4 weeks, and that's it. So I can get some places, but not all places. But the key thing here is that this has this solitary inflorescence, which is like the only thing that's described about lanceolata from the original protologue. So this thing matches fairly well and also fits into the clade where the type specimen would be. So this is lanceolata. These plants, on the other hand, do not have a solitary inflorescence, or they do have this primary inflorescence, but they also have this axillary secondary inflorescence, which has a 3rd collat leaf. And if you didn't notice, the leaves are also alternate, not opposite. This thing is pretty much a stretch to call Claytonia lanceolata in the strict sense. These plants, too, are also currently treated as Claytonia lanceolata, although they do have a name described by Reitberg many years ago in the early 1900s. This is Claytonia obovata, and so I encourage you to go ahead and use that name. It has these very distinctive sunken red veins and heavy betalated pigmentation. The undersides of these leaves are very deep, deep red. And so that brings me around to sort of the questions and past the introduction and getting into the meat of my dissertation and research. And so what I'm going to talk to you today is a little bit about phylogeny. And I'm not actually going to show you a phylogeny, instead I'm going to show you a network, because this is not a bifurcating tree. This is a problem with species groups, is that they really don't fit into the mold that we kind of try and pigeonhole these different taxa into. And so I'm going to talk a bit about relationships and focus more on the Northern California species, but I'll show you all the data that I have so far. And then I'm going to talk about using morphometrics and this application of elliptical Fourier to these outlines of leaf shapes. So this is just a representation of some of the diversity of leaf forms of these different species. And then lastly, I'll touch on how ecological niche varies for these different species, focusing on habitat type in terms of landform and also rock types. And so this is where I started off from. There was a molecular study of Claytonia that was done prior to me, and so this is sort of my launching off point. But as you can see, there's only a single sample of Claytonia lanceolata, which I mentioned earlier, spreads from Canada down to Mexico. And we also understood already that there's quite a bit of diversity in California that has not been sampled. Furthermore, we don't really have any understanding here of how these different lineages relate to each other. So we know there are species, but we don't know how they relate to each other. Thus, I employed next generation sequencing technology. I used double digest RADseq, which is not unlike the AFLP methods that Justin was talking about earlier, except I get sequence data rather than fragment length. And so that's really nice because I can add samples later. But essentially what I did was dove into this black box that we call a genomic data or a read pool, and I did some bioinformatics to transport over to this other garbage bin. And once I got in there, I searched and found some data that I analyzed in a phylogenetic framework. And so this is the network that I'm showing you, and all I really want you to take home from this is what you're seeing is genetic similarity. And so some of these branches here that spread out wide, that's showing conflict where these species might share characters with others, so they're not really fixed for that species, but what you do see is clustering of things that we're calling species. And so I'll just highlight here the Northern California taxa. We've got Claytonia L, which I mentioned was described by Reitberg. We've got this lanceolata species group, probably does have subspecies. I'm not really comfortable with treating any yet, so if it's in California, we're just going to call it lanceolata for now. And then we also have this other species, which is a new species that I'm describing that's restricted to serpentine soils. Also, some interesting things happening here with Claytonia umbellata, which I'll point out now exceeds the scope of my dissertation, so don't ask me about it. This is a real problem child, and effectively it's a tuber with a taproot that extends from below it. So it's pretty much intermediate between the tuberous species and the codico species that I showed you earlier. So I think this is maybe a hybrid species and maybe has formed multiple times. But that, like I said, exceeds the scope of my dissertation. It's an interesting problem that I would like to revisit. But there are some really cool findings that we have been able to find essentially by looking at these relationships and the genetic similarity essentially using this massive data set. And this relationship right here, the Southern California species complex Pearsoni that I focused on most of my research with, is apparently allied with Claytonia obovata. And morphologically, that makes complete sense. These things look very, very similar. Essentially, Pearsoni has these raised veins on the adaxial surface of the cauline leaves, but essentially otherwise, all things are quite similar. But variation within a population is extreme. So if you collected one plant, it might be really, really difficult to key unless you've got a really good example of what I'm calling species. So I wanted to give it this, address this a little bit and see if we really can identify species with high predictive accuracy. And so what I did was apply elliptical Fourier. And essentially, these are leaves that have been cut from a plant, flattened, dried, and then imaged. And I'm just showing you here, we basically use these surgical scissors, get down to the attachment point of the petiole or base of the leaf blade, where it attaches to the stem, and we pull those off, pluck them, flatten them, and dry them. I'm not working with umbellata, partly because it exceeds the scope of my dissertation, but these petioles are really screwy and difficult to deal with. All my other species are sessile, so that's a little bit more straightforward. And to give you just a brief kind of background of what's happening in elliptical Fourier, it's essentially something you can think of as an area under the curve, except the curve is an ellipse. And as you add more ellipses, down here you now have 49 ellipses contributing to the description of the shape of this outline. And so Vincent von Hohm was using beer bottles and whiskey bottles. It's a really interesting paper if you want to read about it. But we're doing the same thing with outlines of leaves instead. So all I'm showing you here is, here's the leaf. We extract an image and then turn it into a binary image, and then analyze those using elliptical Fourier. And the results are best shown in ordination. And so what I'm showing you here is just a pairwise comparison between lanceolata and obovata. I don't have any data for the serpentine species because a late snowstorm came through around Mount Eddy in late April, and basically the plants disappeared under a couple feet of snow in 2015. So I got data for the lower elevation obovata there, but didn't get any data for lanceolata. And if you can't tell from here, there's a clear distinction between the two species, some variation in the leaf shape of lanceolata and obovata that sort of drifts into the morphospace of the other species, but essentially they're categorically different with strong statistical significance. And essentially 95% of the variation is explained by this one eigenvalue, and all you're really seeing along that is a change from these lanceolate, almost linear leaves to these really more rounded, big fat obovate leaves. And so with that, we've got some evidence that supports multiple lineages, both on the genetic level and the morphological level, but what does that mean in terms of the ecology? Are these things actually partitioning different niche space, or are they all occurring in the same place, and if so, what keeps them separate? And so again, I'm kind of going to focus more on the landforms as well as the geology, but there's a lot of other interesting things happening in terms of associated species and different things like that for these taxa. Just to give you a sense of the range of these different species, Serpenticola is the most narrowly distributed of the three, and that's the new species that I'm describing, which does trickle into southern Oregon in the Siskiyou Mountains. But otherwise it's primarily in the North Coast ranges, and it occurs on serpentine soils, although I have found it with shale rocks, and I don't know what those soils are like. So I'm getting soil data, but I don't have any of those data to show you yet. And I'm just highlighting the rocks that obovata grows on because, as you can see, it shares a lot of the same rocks, pretty much all of them, with Serpenticola. So here is Claytonia lanceolata, which I showed you earlier. Probably would fall into subspecies sessilifolium, but as I mentioned before, I'm uncomfortable with treating any subspecies currently for this species, so we call it lanceolata. It's restricted to pretty much felsic, really benign rocks. And it occurs in these more wet mesic environments, anything from a subalpine meadow to the more xeric meadows that you find in the eastern Sierra. Obovata, as I mentioned before, really easy to identify because of this betalain pigmentation character. Is a really interesting species that has these sort of Mickey Mouse ears, which don't really come out in my elliptical Fourier as much as I wanted to see those shapes, because I'm using too many harmonics, or too few harmonics to actually describe the variation. They're very simple shapes, so it doesn't take very many harmonics to actually describe that. And this also shows a wide range of habitats. It can be in these very mesic, sort of almost meadow or meadow-like understory, to these much more xeric subalpine forested habitats. And Serpenticola, again, this is the new species that I'll be describing. And hopefully we'll make it into Florida, Oregon if I submit the manuscript early enough. Has these alternate leaves. It's really easy to identify, and most of the time it has this axillary inflorescence, but that doesn't always occur. And it's pretty much the most xeric adapted. It's on serpentinite and serpentine soils, relatively low elevations to higher elevations. And as you might expect, these two species basically are each other's closest relatives, and what seems to be the reproductive barrier is that they don't occur on the same rocks, whereas these occur on the same rocks but in different portions of the state. But these occur on the same rocks and in the same place, but effectively are the most distantly related out of all of these comparisons. There's a lot of genetic difference between these two, which is really interesting, but effectively you can find these two species high-fiving their colline leaves, which is really exciting. As you might expect, there is clearly some genetic interchange between these, which I can't talk about today, but thankfully the NorCal Bots has given me some money to address this, also using next generation sequencing technology. So I will have a scale up involving basically a hybrid swarm at Mount Eddy, right across from Mount Shasta. But to summarize, just so that I have some time for questions, essentially we're still a little bit confused as to which species are most closely related, and that's in part because of what I think is some hybridization between some of the other close relatives in Claytonia. Definitely species can be distinguished by morphology. I don't know how some of these were missed. Clearly obovata was not. Rydberg got it right. Go with Rydberg. but some cool ecological themes coming out of this where effectively if you're really closely related, you are probably going to be on different rock types because there's too much chance for hybridization to occur, and these things seem to take to hybridization very readily. So there would be a lot of homogenization and effectively no speciation if it weren't for some kind of barrier such as this. So with that, I have a ton of people to thank, collaborators, people sending me material, giving me permits, and other things like that. So just thanks to all of these collaborators especially for helping me out, writing code, things like that. And with that, if you have any questions, I do have time, so I'm open to questions.